Hi class, this is your instructor, Skylar Huff. We have now made it to the fourth chapter, and this is the organization of the cell. The cell is that smallest unit that can carry out all activities we associate with life. When provided with essential nutrients and an appropriate environment, some cells can be kept alive growing in the laboratory for many years. Thanks a lot, people who took cells from Henrietta Lacks. You may hear more about that later. By contrast, no isolated part of a cell is capable of sustained survival. So as you read the chapter, yes class, please have your textbook out, recall the discussion of systems in biology. And this came from the first chapter. So even as we describe individual components of cells, each of which is a system, and of course we discuss how these structures work together to generate ever more complex interacting biological systems within the cell. So the cell itself is a highly intricate biological system, and groups of cells make up tissues, and then there are organs, and ultimately the organisms that are biological systems of ever-increasing complexity. So with that in mind, keep in mind that it's not just one type of cell. There are many different types of cells in existence, such as the prokaryotes, the protists, the fungi, meaning those consist of course, a single cell, not all of which, but some of which do, while most plants and animals are composed of millions of cells. So as it states, most prokaryotes, many protists, and fungi consist of a single cell, and I particularly think of the yeasts in the case of the fungi. In contrast, most plants and animals are composed of millions of cells, and cells are the building blocks of complex multicellular systems. Although they are basically similar, cells are also extraordinarily diverse and versatile. So they are modified in a variety of ways to carry out specific functions. So cell biology is an interdisciplinary science. It draws on increasingly sophisticated tools from a wide array of scientific fields in the quest for better understanding of cellular structure and function. And I can hardly wait to get to these things with you all in the course. For example, investigation of the cytoskeleton, meaning that cell skeleton, currently in active and exciting field of research, has been greatly enhanced by advances in microscopy, biochemistry, molecular biology, genetics, and even, I guess you would say one of my favorites class, computational biology. So the picture micrograph you all see in your textbook found there on page 73. Of course, what it there shows you class is that extensive distribution of cytoskeleton fibers known as microfilaments, and they're here composed of the protein actin, and they're also within tubulin in cells. So the biochemical studies of these 3D images of their protein subunits have led to deep insights into how cells rapidly assemble and disassemble these dynamic complex structures. So the further understanding of their functions come from molecular genetic studies of cells that contain mutations in genes that regulate their assembly and disassembly. So it's through this combination of approaches that scientists are developing a comprehensive picture of how cytoskeleton system maintains shape and functions in cell movement. So with all of this, I say please class make sure you're spending the time to take, I would say, amazing notes. And I say amazing notes because it's here where we really get into the biology of the course. The first chapter we dealt with a view of the scientific process, and that, I guess you say, brief overview of how we have these interwoven systems, meaning with systems biology. And then thereafter we got to chapter 2, where you all learned about basic chemistry, and then of course in the third chapter we got to biochemistry. and even, I guess you would say, along with biochemistry, we looked at organic chemistry. So now, class, in this third chapter, it will be all about the cell. So here we are. As it states, class, here on your screen, that cell is known as, class, being the basic unit of life. So on your exam, be prepared, class, to give these three tenets of the cell theory. Be prepared, class, to be able to exhibit what you've learned about what a cell is. And I'll take my time here. If I go too fast, please let me know. That way, I can go a lot slower. So I am, class, here on page 74. This is section 4.1. And let us now, class, formally begin chapter 4. So to help you with your studies, class, these learning objectives you found at each section of the chapter, these are those things that you should have mastered by completing each section within that chapter. For instance, here on page 74, it states that you should be able to connect the cell theory to the evolution of life. You should be able to also relate the organizational similarities of all cells to the need to conduct essential life functions, and then finally be able to explain the significance, class, of cell size and shape. So let us begin. So a long time ago, class, back in, of course, early in the 19th century, there were, of course, scientists, and those scientists, class, had microscopes. And what happened is there was the botanist class, Schleiden, the zoologist class, Schwann, and a bit later, class, another scientist, Virchow. So it was in 1833 when Schleiden, that botanist, observed things. Not long thereafter, class, the next year, 1839, the zoologist Schwann reasoned that, of course, a plant, an animal, each of which consists of cells. Later on, class, Virchow observed those cells dividing, giving rise to daughter cells. Hence, in 1855, Virchow proposed that new cells form only by the division of previously existing cells. So, what I'm stating, class, is this. These scientists, Schleiden, Schwann, and Virchow, they contributed, class, to the development of this cell theory. And I'll make it even more plain now. The cell's theory states that, class, cells are the basic unit of life. You should write that down. Secondly, all living things are composed of cells, or at least one cell, of course, in the case of bacteria. And thirdly, class, all cells arise from pre-existing cells. So with those three things in mind, there is evidence, class, of such. So you can go back to ancient times, and it's evidence, class, that all cells have that common origin, and it's provided by those basic similarities in their structures and in the molecules in which they make them up. So when we examine that variety of diverse organisms ranging from, class, just bacteria, yes, I say simple bacteria, being unicellular, to the most complex plants and animals, we find those striking similarities at that cellular level. So it's those careful studies of shared characteristics, class, that help us to achieve that evolutionary history of various organisms, and of course they provide that powerful evidence, class, that all the organisms of life today have a common origin. So since cells are that basic unit of life, meaning cells arise from pre-existing cells, and that's where life begins, it's the very same, class, that cells are able to maintain homeostasis. And as I say this, they must have that appropriate internal environment, class, be maintained, meaning maintenance of that appropriate internal environment. And that is necessary for those biochemical systems to function. So for the cell to maintain its internal composition, class, those contents must be separated from that external environment. So this gets us, class, to one of those major portions of all cells, and this is known as that plasma membrane. So it is a structurally distinct surface membrane that surrounds all cells. So it makes glass the interior of that cell, that enclosed compartment, and that plasma membrane allows the chemical composition of the cell to be different from that outside the cell. So it's this plasma membrane that has those unique properties that enable it to serve as that selective, I say again, that selective barrier between cells' contents and the exterior, permitting class the cell to exchange materials with the environment and accumulate substances needed to drive its biochemical reactions. So if you think back with me, class, to our prior chapter, I mentioned that plasma membrane is amphipathic, being selective, letting only these things in and not those things. I'll continue. Next up, class, is that cells have internal structures called organelles. So with these organelles, class, I'm stating that they are specialized to carry out cellular activities. And it's the case that most of the organelles of eukaryotic cells, meaning a eukaryotic cell, class, is a cell that possesses, I repeat, a eukaryotic cell is a cell that possesses a nucleus, and they consist of one or more membrane-bound or membrane-enclosed compartments. They're called organelles, class, and they consist of one or more, of course, of those. They regulate, class, their very own environment for specialized functions, such as converting, class, energy to usable forms. And I'm referring, class, to the mitochondria when I say that. So they also, class, process nutrients. They recycle damaged or unneeded structures. And as I think of an organelle, class, that would recycle those damaged or unneeded structures, I would think of the lysosome. That is the role of lysosomes. And they also, class, take our genetic information that is stored in DNA molecules, which you all should remember now. That's deoxyribonucleic acid. For example, is contained. It's also going to be duplicated by the process of replication and transcribed by the process of transcription in the nuclear compartment of eukaryotic cells. So what I'm getting at is these organelles, class, serve a variety of functions that make you and me who we are today. So lastly, I'll get to the cell's membranes. They also, of course, organize the surfaces for interacting proteins. So this way, the cell can communicate and even transport things by use of those proteins. So our cell, class, cannot be too large, and of course, it is not efficient if it's too small. So the organization of the cell and their small size allows them to maintain homeostasis well. So most components of the cells are measured, class, in nanometers. In lab, we got to the micrometer. Well, now, class, we're down to the nanometer. And I'll put it this way. 1 micrometer is equal to 1,000 nanometers, because I think it helps to get an idea of what I mean as far as size. So, having stated that, I'm just going to move on over to the way in which our cells, class, must remain small. So the question is stated in your textbook, class. You'll see this on page 75. Why are most cells so small? So if you consider what a cell must do to maintain its functions and grow, class, it must be easier to understand the reasons, firstly, for its small size. So a cell must take in food and other materials and rid itself of waste products generated by those metabolic reactions. Everything that enters or leaves the cell must pass through the plasma membrane, class. So the plasma membrane contains specialized pumps or channels with gates that selectively regulate the passage of materials into and out of the cell. So the plasma membrane must be large enough relative to the cell volume to keep up with the demands for the passage of materials. Thus, a critical factor in determining cell size is the ratio, class, of its surface area, that plasma membrane, to its volume. So with this, as the cell becomes larger, class, its volume increases at a greater rate than its surface area, or plasma membrane, which effectively places an upper limit, class, on cell size. So above that critical size, the number of molecules required by the cell could not be transported into the cell fast enough to sustain its needs. In addition, the cell would not be able to regulate its concentration of various ions efficiently to export its waste. So I'm saying all of this to hopefully ensure that you know, class, that cells, of course, are small because they must maintain that larger surface area than volume, and the ratio is critical of which. So this allows in class to efficiently transport those materials into and out of the cell, maintaining homeostasis. So from here, I'll move on over. And it just mentions that cell size and shape are adapted to function. So the amoeba and even the white blood cells can change shape as they move to, of course, engulf things, such as engulfing the non-self or engulfing class something to ingest for food. Sperm cells have a long whip-like flagella. And of course, the flagellum allows the sperm cell to move, providing locomotion. Nerve cells in the converse class, they have those long, thin extensions that enable them to transmit messages over great distance. Those long, thin extensions class, at least for transmitting messages, are known as the axon. For instance, I guess I'll go quickly here and draw a quick neuron. So what I'm drawing here, class, would be, well, a neuron. All right. So here, class, those are known as dendrites. That's an arrow. Those are dendrites. That's an ugly arrow. Better arrow. So the dendrites class are that receptive region of the nerve cell. They take in those impulses. So within this portion of this neuron is the cell body, and that's the nucleus. The long extension class I just mentioned is there, the axon, and it transmits class those messages in one direction. And that's the direction in which class those nerve impulses are transmitted over those great distances, up to even a meter long class in some people who are quite tall. And then we have those rectangular epithelial cells that stack like building blocks to form sheet-like tissues. So this class is that difference we have, or at least a few of those differences we have in the size and shape class of our cells, which are quite adapted to their function. So to study cells class, I'll go over this a bit briefly. But in the late 19th century class, I hope it's now obvious that microscopes were, of course, developed so that biologists could study those cells, and they could study them efficiently. And as this happened, class, it was Robert Hooke that first described cells back in, of course, the 17th century, meaning 1665. And he made that microscope. I would imagine that would have been a pretty amazing microscope to see these days, to see that first microscope, or at least what is known as that first microscope. And then Leeuwenhoek class discovered bacteria. I say discovered bacteria. That should be mind-blowing, but it may not be. But I'll say it again. Then Leeuwenhoek came along class and discovered bacteria, protists, blood cells, and even sperm cells. And among the first scientists class to report cells in animals, Leeuwenhoek was. So he was a merchant who was not formally trained as a scientist class, but his skill, curiosity, and of course his diligence in sharing his discoveries with scientists at the Royal Society of London brought an awareness of microscopic life to the scientific world. So unfortunately, Leeuwenhoek did not share his techniques. At least, it was not until that approximately 170 years later class that, of course, that there were microscopes for the biologists to use, as previously stated. So the light microscope class, what you all will use in lab. This is here class. And what happens of which is you just have that tube with glass lenses at each end for which visible light passes through the stained living cells. So the lenses will refract and then magnify the image. This is showing you that coarse depiction of a light microscope, much as you see in lab with your arm and base. But more importantly, class, the light microscope is here for your eye to look inside. So as your light looks inside, as your eye peers inside for you to look inside, what you're then seeing, class, is from the light source located here, shining that light, and it goes through lens, until ultimately, class, you're able to see what's there. So magnification is that ratio of the size of the image seen with the microscope to the actual size of the object. So in terms of magnification, class, I would just say that the best light microscopes magnify an image, class, to no more than 2,000 times. And the light microscopes we have here at the school have a total magnification of 1,000 times. So with this, in terms of resolution or resolving power, it's just that minimum distance between the 2 points at which they can both be seen separately. And that just depends on the quality and wavelength of light. And it increases, class, as wavelength decreases. I won't go through each of these types, class, of microscopy. However, I'll do just a bit. So bright field microscopy, class, of course, using the light. And shown to you all here is, of course, from your textbook on page 77, class, figure 4.3. So using a microscope, Robert Hooke looked at a thin slice of cork and drew what he saw. That's there at A, which you all cannot see at the moment. At B, class, you're seeing, of course, bright field microscopy, and that, of course, is just a stained image. Dark field microscopy, class, is shown here at C. And at D, you can have a phase contrast, and finally, class, here at F, that's a confocal image, showing you the plasma membrane and the contractile vacuoles that are stained red, and even the proton pumps stained yellow. Electron microscopes, class, are used to study ultrastructures of cells. So some electron microscopes have a resolving power, class, of less than 1 nanometer, and that's quite amazing. So the electron beam consists of energized electrons focused by electromagnets. So there is a transmission electron microscope, as well as, class, a scanning electron microscope. So it really does depend, of course, on which of those types of microscopes, of course, you're using. So the electron microscope came into wide use in the 1950s. And from then until now, class, that microscope has changed, I would say, greatly, dramatically even. And cell fractionalization, or at least fractionation, is that technique, class, for separating parts of cells for studying. So I won't spend a great deal of time with this, but after you, of course, have broken up the parts of that cell, you centrifuge it to separate those parts, to extract it, class, into a supernatant, meaning that pellet and the supernatant. And then, of course, you can centrifuge the supernatant again, and, of course, into another pellet for differential class centrifugation. So the pellets can be resuspended or the components further purified by the density graded centrifugation. So this is to see those parts that were in the cell and, of course, separate them, class, from those other parts of the cell. So now, class, we've finally made it to the 2 basic types of cells. And I say it that way because this is where we get into, of course, I guess you say, the biology, the cells. And yes, I'm smiling. Let us begin. So there are 2 basic types of cells. The first type, class, is the prokaryotic cell, and the second type is the eukaryotic cell. Having said that, keep in mind, class, that it's bacteria and archaea, or you can call those, class, archaebacteria. And, of course, those are prokaryotic cells. So all other known organisms, class, I repeat, all other known organisms consist of 1 or more eukaryotic cells. And I say that should, class, be a quick and easy way, class, to divide up those 2 basic types of cells. So here on the left, class, is a prokaryotic cell. That's that typical bacterial cell, class. And, of course, you see right there that flagellum. And then on that right-hand side, class, that would be a protist. That's a protist indeed, showing you an example of what a eukaryotic cell would look like. So continuing on here, with bacteria and archaea, or archaebacteria, let us now begin. So these types of cells are typically smaller than that eukaryotic cell. So yes, these are prokaryotes here, the prokaryotic cell. So I say again, these cells are typically smaller than the eukaryotic cell, and that on average, they're about 1/10 of the diameter class of that average eukaryotic cell. So in prokaryotic cells, the DNA is typically located class in that nucleoid region. I repeat, in the nucleoid region. Unlike the nucleus class found in eukaryotic cells, that nuclear area or nucleoid class is not enclosed by any membrane at all. So that term prokaryotic class also means pre-nucleus or before the nucleus, and it refers class to this major difference between the prokaryotic and eukaryotic cells. So other cell types, many other types of internal membrane enclosed organelles, are also absent class in prokaryotic cells. And I'll say it this way, class. Prokaryotes, by definition, have no membrane-bound organelles. They have no membrane-enclosed organelles. And class, if you can learn this, there is no question I can ask you in the test about, of course, contrasting these two types of cells that you cannot answer. So up next of which, like eukaryotic cells, prokaryotic cells, they do have a plasma membrane. All cells do, class. That surrounds the cell. So the plasma membrane confines the contents of the cell to an internal compartment. So in some eukaryotic cells, the plasma membrane may be folded inward to form that complex of course, membranes, along which many of the cells' metabolic reactions take place. And most prokaryotic cells do indeed, class, have a cell wall. Most do. And those cell walls are, of course, extracellular structures that enclose that entire cell, including the plasma membrane. And many prokaryotes, class, have a flagellum, or they have flagella. So flagella is plural, and flagellum is singular. So those are long fibers that project from the surface of the cell. So it's that prokaryotic flagella that operates like propellers, class, and they're important in locomotion. So their structure is different from that flagella class found in eukaryotic cells. It's very different from that found in class eukaryotic cells. And some prokaryotes, class, also have hair-like projections known as fimbriae. And fimbriae, class, are used to adhere, class, to one another or attach to cell surfaces of other organisms. Next up, class, I'll get down to what else is inside. So the dense internal material of the bacterial cell contains ribosomes. Yes, so both prokaryotes and eukaryotes, class, have ribosomes. And those are the so small complexes, class, of ribonucleic acid, or rRNA, and the protein class that synthesizes polypeptides. And I hope you remember from last chapter, class, that you can refer to polypeptides as, of course, proteins. So the ribosomes of prokaryotic cells, class, are smaller than those found in eukaryotic cells. Keep that in mind. They both have ribosomes. It's just that those in prokaryotes are smaller than those in eukaryotes. And of course, the prokaryotic cells contain storage granules that hold glycogen, a lipid, or phosphate compounds. So let us now continue. Here, class, is a figure from your text. You'll find this, class, on page 83. I guess I'll try to go in the same order we went through as we began this there. So if you look closely, class, this is the nucleoid or nuclear area. So I might just draw it and put maybe a little line here, just like that. So you know that is what contains their large circular chromosome. From there, of course, it does say that there is DNA. And next, I believe, was the cell wall. The cell wall looks a bit brown here in this photo. It looks brown in that photo. And then just inside that cell wall, class, which is orange, is the plasma membrane. The plasma membrane. Up next, class, I think you can definitely see this long flagellum here. The long flagellum. I just outlined the flagellum. And then, along with that, class, in red are the ribosomes. The red ribosomes. And of course, kind of a purple color, or even a bit purple, are storage granules. Maybe it's swatched within. And one thing I have not got to yet, class, are the embryae. The embryae. And one thing that I did not mention at all, class, is the capsule. The capsule that encloses everything. And some have a capsule, and some do not. It just depends, class, on that bacterium. This, class, is that structure of the typical prokaryotic cell. Study it, please. So from here, I'll move on, class, to the other type of cell. This, class, is the eukaryotic cell. So, by definition, class, eukaryotic cells, they're defined by the presence, class, of membrane-bound organelles. I can't say it any other way. So, eukaryotic cells, as it states in your text on page 83, class, are characterized by highly organized membrane-bound enclosed organelles. It includes, class, that prominent nucleus, which contains the DNA. And of course, by now you may know it. I hope you know it for sure because you've been studying. But it's DNA, class, deoxyribonucleic acid, that of course is the hereditary material. So the term eukaryotic, class, means true nucleus. And when we get to, of course, the area outside of the nucleus, it's referred to as being the cytoplasm. We've already finished up, class. The nucleus contains DNA. Well, the cytoplasm, class, is that part of the cell that, of course, I guess I'll say that is outside the nucleus, and even, class, within the nucleus itself, called nucleoplasm. But I'll just refer to, class, as cytoplasm, not so much nucleoplasm. And there are various organelles, class, suspended within that fluid component of the cytoplasm, which is called cytosol. So I'll say again that the area of the cell, class, outside of the nucleus is referred to as a cytoplasm. And of course, those organelles are suspended within the fluid component of the cytoplasm, which is known as the cytosol. And I won't try to be as, I guess I'll say, specific as those were. Just make sure, class, you pay attention to that. So eukaryotic proteins are synthesized in the cytoplasmic compartment, class, by ribosomes. I say it again. Ribosomes, class, are here to synthesize proteins, synthesize polypeptides. So those large macromolecular structures can then, of course, function as components of that cytosol when they produce soluble proteins. Or those be firmly bound, class, to the cytosolic surfaces of the membranes and form the proteins that are either attached to that membrane or enclosed in compartments, class, bound by the membranes. So to make sure you don't get confused, class, what I meant about cytoplasm and cytosol, check this thing. In your text, it just states that cytoplasm, class, includes both the cytosol and all organelles other than the nucleus. That's why it's sometimes referred to being, all right, that's cytoplasm, whereas the cytosol, class, is, of course, just that fluid. So here, the many specialized organelles of the eukaryotic cells solve some of the problems associated with that large size. So it's the eukaryotic cells that can become larger, class, that are larger than the prokaryotic cell. So eukaryotic cells are different from prokaryotic cells in having a supporting framework, that cytoskeleton I mentioned, class, to start the chapter, that's important in maintaining the shape and transporting materials within the cell. So some organelles are present only in specific cells, and the example being, class, the chloroplast. So the chloroplasts, the structures that trap sunlight for energy conversion, class, are only in cells that carry out photosynthesis, such as certain plant or algal cells. Cells of fungi and plants are surrounded, class, by a cell wall, and that cell wall is, class, external to the plasma membrane. So plant cells also contain that large membrane-enclosed vacuole. So there are just some things that are unique about this cell and that cell. And in that, of course, only a certain type of cell has that structure. So the membrane compartments class allowed the eukaryotic cells to carry out a diversity class of activities. So from this point forward, class, I'm going to prepare you better, I say, for your lecture test. And I'm just going to go through eukaryotic cell structures, because on your lecture test, you'll be tasked with, of course, describing both structurally class and functionally 9 eukaryotic cell organelles. I say on your lecture test, you all will be tasked with describing 9 membrane-bound organelles, or at least 9 organelles, structurally class and functionally. So if you have a good bit of paper left, great. If you don't have much space left, I'll say get more space, and we'll begin in about 4 seconds. All right, so in describing class, keep in mind that you won't just describe those eukaryotic cell organelles. What you will also describe, class, is the ribosome. Yes, it's not a membrane-bound organelle. However, you, of course, will describe it. So I'll begin, class, with what is before you called the cell nucleus. So I'll go with that structural description first. So it's a large, double membrane-bound organelle containing, class, the nucleolus and chromosomes. In function, class, it stores information as deoxyribonucleic acid, meaning it stores it stores hereditary information, class. So DNA is transcribed into RNA, and then thereafter, class, RNA synthesis is what goes on to specify the proteins. This has been, class, the nucleus. And one thing I'll mention, class, is that there are nuclear pores. Those nuclear pores, class, are there, and that's what allows the RNA to leave the nucleus, class, and of course enter into the cytosol for protein synthesis. I'll continue. Next up, class, it just mentions DNA replication. I will spend hardly any time here because we'll get to this, class, in a later chapter. But DNA replication happens by way of replication, and this happens because cells divide. And as cells divide, class, going from one cell to 2 cells, you must have, class, I would say, enough genetic information, class, for 2 cells, as opposed to just having the DNA, class, in one cell. One other thing I'll mention, class, about DNA is that this is, of course, where our genes are located. And yes, each cell is unique, so that it's not that every gene is turned on in every cell. However, the information, class, for all genes are in all cells. So to stress once more, class, it is within the nucleus that transcription occurs. And of course, it's transcribing what's in DNA into what's, class, then referred to as mRNA, and this, class, is what specifies proteins, as of course the RNA leaves the nucleus and of course moves into that cytoplasm for translation to occur. Take notes well. So I'll mention chromatin, class, here briefly. Chromatin, class, is that state of DNA that's, I guess I'll say, synonymous with noodles. I did say noodles, such as a bowl of noodles or oodles of noodles in a bowl that have been boiled already, not the ones that are dry. So DNA associates with RNA in that chromatin state in certain proteins. So this, class, is how it's packed in the nucleus. And then for division of cells, the chromatin, class, is condensed into that chromosomal state. And in us, class, there are 46 chromosomes, 23 pairs, meaning half of this, class, 46 came from mom, and half of that, class, 46 came from dad. Whoever they are, whether you know them or not. That's how it happened. And then, of course, we have that approximate, class, yes, 2 meters of DNA. That's a lot. Billions of A's, T's, G's, and C's. So it's, class, the nucleolus that is the site of RNA synthesis, as well as, class, ribosome assembly. And you will not describe it in your test. I'm moving on from the nucleolus, class. This is it, class, that eukaryotic cell nucleus. So if there are questions, let me know. Otherwise, I'm moving. The ribosome. So the ribosome class is not a membrane-bound organelle. However, you will still describe this class both structurally and functionally. In structure class, the ribosome has granules composed of course, ribosomes and protein. So some class are free and some are attached. Those that are attached class are attached to the endoplasmic reticulum, and those that are free class are free in the cytosol. So it does class have a large subunit and small subunit. So in function, meaning I gave you the structure, in function class, ribosomes synthesize proteins. They synthesize polypeptides in both eukaryotes and prokaryotes. So with this endomembrane system, I won't mention it at this moment, class. I'll spend more time with what I just mentioned, meaning the endoplasmic reticulum. So to describe it as a whole, I'll say it's a network of internal membranes that extend through the cytoplasm. So functionally, the cytoplasm class, functionally, the endoplasmic reticulum, it synthesizes lipids. That means to create lipids. And it also modifies many proteins. So this is, I would say, that origin of those intracellular transport vesicles that carry proteins. So to get more specific here, class, I'll begin with that called the smooth endoplasmic reticulum. The smooth endoplasmic reticulum class in structure lacks ribosomes on the outer surface. Hence the name, class. It's smooth. Functionally, class, what it does is goes through the process of lipid synthesis. That's one. Drug detoxification. That's two. And then, of course, it's there for calcium ion storage. I say again, the smooth endoplasmic reticulum class stores calcium, detoxifies drugs, and there to synthesize lipids. The rough endoplasmic reticulum class is in structure, studded with ribosomes on the outer surface, and functionally class, it is here to synthesize proteins or manufacture proteins. And it's, of course, those proteins that will leave the cell. But we'll get back to more of that later. This class shows, so I kind of put a dividing line here. On the left, class, this is the rough ER. On the right, class, that is the smooth ER, endoplasmic reticulum. In red, class, those are ribosomes. Studied. Up next is the Golgi Complex. Structurally, class, they are stacks of flattened membrane sacs. Functionally, class, the Golgi Complex packages secreted proteins. It packages secreted proteins. I mean, these are the proteins that will eventually leave the cell. So I'll think about sometimes the packages received at the house I live at. Sometimes they're delivered by the United States Postal Service. Other times they're delivered by FedEx. And even others, they're delivered by those who are brown call, those for UPS. You can kind of think of the Golgi Complex as being the same. And you say, what do you mean by being the same? Well, they sometimes say that it's logistics, meaning the Golgi Complex class, it sorts proteins, modifies proteins, and of course secretes those very same proteins. So that's the Golgi Complex. And please don't write down, class, UPS on your test. That would be incorrect. So each stack class of Golgi has a cis phase for entry, a trans phase for exit, and that medial region class, that's in between. Thanks a lot, modification. Next up, class, a figure showing such. So yes, I said that each of those phases of Golgi class are just a bit different. And the difference there, class, is of course for function. So as you look at the figure and the arrow I've drawn, class, for Golgi, hopefully you all can see and kind of get an idea when I get to the differing phases there. So that cis phase is their class for entry. Thanks a lot, what just happened, class, by way of our rough endoplasmic reticulum. So the medial region class is that is in between. And then you have, of course, that trans phase for the exit class. Those proteins that came in to be sorted, the ones came in to be modified. And then ultimately class those that are secreted class, still of course leave the cell. So I can't stress enough. I've said this, I think, twice. Here's my third time, class. That typical sequence followed by that glycoprotein, destined for secretion from the cell. The polypeptides class are secreted on the ribosomes. And what had happened was, I did just say that, by way of your DNA. Your DNA class says, well, excuse me, I won't say your DNA said it. I'll say by way of cell communication class. It was known that, of course, that glycoprotein was needed. So the gene for that glycoprotein class was transcribed from DNA to RNA. That mRNA class then left the nucleus by way of those nuclear pores into the cytoplasm. Of course, the mRNA then class went to a ribosome. Ribosome class then, of course, translated the protein. The protein class was assembled and the carbohydrate portion was added in the lumen class of the endoplasmic reticulum. So what then happened is that rough ER class transported that protein, the glycoprotein class, to the Golgi, or the cis phase. It began to be modified class. And then thereafter, in the trans phase, the glycoprotein is in package class. Once the glycoproteins are packaged into the transport vesicles, they'll be transported through the plasma membrane and then ultimately exported from the cell by way of exocytosis. Next up are lysosomes. Structurally, the lysosomes are membranous sacs. Particularly, you find lysosomes class in animals. Functionally, lysosomes contain enzymes that break down ingested materials, and they also break down damaged or unneeded organelles and proteins. So they contain that estimated 40 types of digestive enzymes, and within the lysosome itself, they have a pH about 5. So with all of that, just ensure that we'll get to a lysosomal storage disease later on, and that's called Tay-Sachs disease. We'll do more of that once we get to a bit of genetics. So within which, just keep in mind that sometimes that there may be one or more primary lysosomes that will fuse with a vesicle when the bacteria or degree is engulfed by a cell, and that secondary lysosome will help to, of course, digest that cell. Next up are the vacuoles. So vacuoles are mostly found in fungi, plants, and even algae, and they are structurally classed membranous sacs. So what the vacuole does is store materials, store wastes and water, and ensure that the hydrostatic pressure class is maintained within our cells. And that class is especially the case in plants. So I say in plants because in plants they must have that turgor pressure. Their cells must remain turgid, and it's as much as 80% of that plant cell's volume is nothing more than this vacuole, and that's a lot of it. So when I say they're turgid, that means they have that high pressure. And from here, we'll keep moving on to the next, being the peroxisomes. The peroxisomes are two structurally classed membranous sacs, and they also contain a variety class of enzymes. So in function class, the peroxisomes are the site of many metabolic reactions that break down fatty acids. That's one thing. The peroxisomes also class synthesize phospholipids, and they even class degrade the alcohol in yeast cells, which I find amazing. They also function to detoxify compounds in our liver and kidney cells. So they're majorly important class in the detoxification. And then lastly, class, the peroxisomes can convert stored fats into sugars in plants' seeds. And I love seeds. So since we are here in the peroxisomes, let's get to, of course, further things about what they do. So our peroxisomes are named as such because of the fact that they go through those oxidation reactions and they produce what is known as hydrogen peroxide. It's H2O2. Well, in that hydrogen peroxide actually class detoxify certain compounds. So, of course, but this compound, if it were to escape the peroxisome class, it would damage other membranes in the cell. I mean, it's pretty dangerous. In fact, I say toxic. So what then happens, class, is that peroxisomes contain the enzyme catalase. So I'll draw this line here, and that should be an arrow. And I'll write down here catalase, and for those paying attention, it's typical class of enzymes to have that suffix being ase. So what happens is catalase class rapidly splits that excess hydrogen peroxide, that H2O2, into, of course, what is water class and oxygen. So, of course, what we'll be left with is, I guess I'll write this equation as being there are two H2O2. And we have 2H2O plus O2. And time that's mean, let's keep moving it right along. So the mitochondrion is next. So we have structurally class. The mitochondrion is, of course, those sacs consisting of two membranes. So there is that inner membrane that's folded class to form cristae, and of course within which it encloses the matrix. So now that we've gotten that much done, I'll show you, class, here on the left-hand side, class, the mitochondrion. So you see, class, those two membranes that are there. So this, class, the mitochondrion, is the site, class, of most reactions of cellular respiration. So that is that transformation of energy originating from glucose or lipids into ATP, that molecule of energy. So what happens in this case, class, is I say you must eat. First things first, eat. Provide your body with glucose. Of course, we take in oxygen, class. Well, from, I say, just those two things, that should be a number 2. I'm trying to write a number 2 there. From just those two things, class, we synthesize adenosine triphosphate, ATP. And as a byproduct, class, CO2 is given off in a bit of water. This, class, is what allows us all to live, meaning without this process, life wouldn't occur. Life would not exist. Secondly, class, are the chloroplasts. So chloroplasts are also a double membrane bound structure enclosing that internal thylakoid membrane. So the internal thylakoid membrane, we have those stacks, those flattened stacks. And I'll leave it at that for now because I'm not teaching photosynthesis. But what I will say also structurally is that the chloroplasts contain the pigment chlorophyll in the thylakoid membrane. Hence, of course, the color green. The chloroplasts, or plastids as some may call them, are the site of photosynthesis. So chlorophyll captures light energy and, of course, along with that, class, the ATP and other energy-rich compounds are then produced to then convert, class, carbon dioxide to carbohydrates. So yes, light strikes the chloroplasts, and of course the chloroplast is there, thylakoid membranes, CO2 class enters into that leaf, water class comes up by way of the roots, thanks a lot, soil and rain. We get ATP here, class, and not just ATP, but there's another molecule of energy which I won't mention here. And then from such, class, water is actually going to be split. So as water is split, class, oxygen is given off as a byproduct, and ultimately, class, glucose is synthesized. Talk about life, class, that is how life occurs. So this is by way of the process, class, of aerobic respiration, that chemical energy, class, is, of course, going to be synthesized, meaning converted, class, the chemical energy, into that synthesized ATP I just mentioned. So yes, we have that double membrane bound class, organelle, and of course we have that inner and outer. So apoptosis, class, it mentions, is important because by way of apoptosis, this is the way in which, class, those cells we don't need, that tissue we don't need, class, allows that tissue to be destroyed, class, provide us, class, with fingers. And of course things are not webbed. But can apoptosis, class, initiate apoptosis by interfering with the energy metabolism or by activating, class, destructive enzymes? So as it's stated there about apoptosis, one thing I like to think about, class, with apoptosis it mentions class cytochrome C. That's incorrect. It should be cytochrome. Cytochrome C is what activates those caspases, which cut up those vital compounds in the cell. And as it happens, class, it's the mitochondria that initiate apoptosis or cell death. And it does it, class, in different ways. So it's not just one way that it happens. But it definitely, class, is by way of cytochrome C, which is a protein that is their class and important in energy conversions. So we'll get to that just a bit, class, when we get to the transport chain, the electron transport chain, that is. So one thing I'll get to is that there could be a mutation class. Well, I'll get plural, say mutations class in of course, the DNA called mitochondrial DNA. And that could be a reason, class, that there are certain genetic diseases in existence. And with that, it's all because of, well, those free radicals. I mean, the free radicals are toxic. And it likely, class, relates directly back to those free radicals. So up next, class, it mentions the pigment class chlorophyll. This is what traps the energy for photosynthesis to occur. I've given you all of this already, so I'm going to keep on rocking. All right. So from here, we'll get down to the structure. So this class shows that outer membrane, and yes, you now, I hope, see the inner membrane located right there. The outer membrane, I just mentioned, class, is there. And I mentioned the thylakoids earlier. I kind of drew lines like this. Those are the thylakoids, class. And of course, a granum is a stack of thylakoids, as your text mentions. And you have the intermembrane space, which we'll do a lot more, class, once we get to this chapter for study. We're not quite there yet. The chapter, class, pretty much will end with the cytoskeleton. I won't spend a lot of time here, but one thing, class, and you will not describe these in the test, would be, of course, the microtubules. These are hollow tubes made of the protein tubulin. They're here for structure support, class, for the cell and cell organelles, and even, class, movement by way of cell division. And this includes, class, its components of being in cilia, flagella, and centrioles. So as we are here, nextly, I guess I'll get down to the cilia and flagella. I'll just draw a very quick cell, and I would say this way. These, class, would be how cilia appear to look. And I guess I could have drawn maybe one of these cells and called this maybe a pseudo-stratified ciliated columnar epithelial cell. I said a pseudo-stratified ciliated columnar epithelial cell. These would be the cilia there, moving some substance, class, likely in that direction, or moving some substance, class, likely in this direction. As opposed to, class, what you are about to see when I draw the flagellum. So let's just draw this portion that's here, put that portion that's there, and that portion that's there. So what I've now drawn for you, class, is that whip-like flagellum. So that flagellum, class, is there for the movement of that cell. In particular, class, I would say that sperm cell. I'll put here in because sperm cells, class, are HAPLOD, called haploid. That's a lowercase n, meaning it has 23 chromosomes, meaning one complete set of chromosomes instead of two complete sets of chromosomes. Hence, that cell is haploid. Up next, class, microfilaments. So the microfilaments in structure are solid, rod-like, and of course made of protein, the protein actin. And with that, class, the microfilaments provide structural support to the cell and cell organelles, and even movement, class, in cell division, and of course, movement of organelles. In muscles, class, they have two specialized microfilaments, and they are actin and myosin. So those are the contractile proteins. So it's those two that are their class that allow contraction to occur. Without those two contractile proteins, there will be no contractions. So nextly, it mentions here, class, the intermediate filaments. So we'll get to centrioles later on, and we've already done cilia, and of course the flagella. So the cell coverings, class, this mentions the glycocalyx. And I'm kind of getting to that extracellular matrix and what just gives our cells, I guess, that covering and its strength. So to kind of end this thing, meaning with the extracellular matrix, class, it's just that many animal cells are surrounded by an extracellular matrix, which they secrete, and it just consists of a gel of carbohydrates and fibrous proteins. And that main structural protein, class, is collagen, that forms many of those tough fibers. So this has been, class, chapter 4. This chapter was longer than prior chapters. However, class, I would say the amount of content is about the same. So make sure you all, class, study quite well, knowing your cell theory. Make sure you all know that difference between the prokaryotic and eukaryotic cells, and can go through and describe, class, both structurally and functionally 9 eukaryotic cell organelles, along with, class, the ribosome, which is not. This, class, has been your instructor, Skylar Huff. I thank you all for listening. Prepare well, take notes, and study well.